Hello students, this is Faizan Mirza. We are discussing transport in plants. Now we'll be focusing on the distribution of various tissues, what we already have discussed in the part 1 of this series of this lectures. The distribution of the tissues in various parts of the plant. So let's start our discussion with the distribution of these tissues in a stem. So when you look at a stem of a dicot plant, so this is the part of this stem section which actually you are looking through a microscope, a light microscope at 60 magnifications. You can see that this layer is the epidermis and this is the cortex region and this is the pith. Epidermis is a single cell lining. You can see the single cell structure here. This entire is the epidermis. These cells in the initial part, these have additional strength because of the cellulose which is deposited here. So these cells will be of the collenchyma. And these larger cells which have intercellular spaces in between, these are the parenchyma cells. So your cortex of the stem is having the epidermis is before the cortex and the cortex is then having your collenchyma and the parenchyma. This is followed by the pith. If you draw a plant diagram, so you draw a plant diagram using straight lines without any breaks to show the distribution of these tissues and the relative width which is occupied by that tissue. Here we are not distinguishing in this particular slide that which part is the collenchyma and which one is the parenchyma. So you have, because this is a bit difficult to see here, but you can see the intercellular spaces very well here in between these cells which are the parenchyma cells, and the same kind of intercellular spaces are not very well here, and these are a bit smaller of the cells, probably because of some air spaces which are much larger, or probably because of the damage done to the damage done, because this is some damage because of the artifacts. These artifacts are added to a slide when the slide is not made properly. So what is this? This is a vascular bundle. So this entirely is a large vascular bundle. These two are tiny vascular bundles, and these are just part of these vascular bundles. So in this vascular bundle, you can see 4 different kind of tissues. You can just use your observation skills to see that these are staining together. This stains differently. These stain differently, and this stains differently as well. So this region is the fibers. These are the sclerenchyma fibers, followed by the phloem. This is the phloem, and this region is the cambium. So you draw cambium with a very thin structure, and this entire is your xylem. So this is how you can just probably make an outline of how these parts are drawn. And if you are drawing a plant diagram, so your plant diagram should have the details of, this is a low power plant diagram of how the vascular bundles tissues they actually look like. So you have sclerenchyma fibers, then you have the phloem. I've drawn this one, and this is your phloem, then cambium, and then you have xylem. These are the air spaces which you can see between the parenchyma cells. So if in this slide, in this diagram, you can see again these parts: the sclerenchyma, the phloem, the cambium, and the xylem. And this thick lining is the epidermis here, and then this is the cortex cells, which are collenchyma, and this is then parenchyma of this tissue, and then this is cortex, and then this is the pith. So these are the parts of a tissue of the stem, how these tissues are distributed in the stem. This shows to you the distribution of tissues in the root, and here you can see larger air spaces in this root. So you draw a plant diagram accordingly. You show these close regions as they appear, because these are not the artifacts now. These are actually the closed spaces. These are the spaces, air spaces through which air can move in and out of different parts of the tissue of the roots. So this is the epidermis here, the outermost lining, and this is the cortex, and here you have the endodermis. This circle is endodermis, and then you have xylem, and this is phloem, and just beneath the under the, you can say, the endodermis, and before the vascular bundle, you will have the cells of 1 to several cell thick layer, and this is pericycle. So this is how these tissues are distributed, and we already have seen in the previous section of the lecture series in part 1 what parenchyma is, what endodermis is, what xylem was, phloem, what pericycle was, what's endodermis. So when we use the term endodermis, pericycle, phloem, xylem, air space, or whatever, so this should just rightfully ring bell in your mind that what part of tissue this is, or what features generally this tissue should be having. That's all from my side. That's it. Thank you so much.